Chapter 6, Topic 1, Microbial Nutrition. In this topic, we're going to be discussing microbial nutrition terms. We're going to talk about what microbes eat, essential nutrients, transport mechanisms, and osmosis. These different topics will help us understand what we need to be able to grow microbes in the lab. Conversely, in environments where we don't want microbes to grow, if we know what they need to grow and remove those from the environments, then we'll be able to inhibit the growth of microbes. The learning objectives for this topic are to be able to list the essential nutrients of a bacterial cell, be able to differentiate between macronutrients and micronutrients, be able to construct 4 different terms that describe an organism's sources of carbon and energy, be able to define saprobe and parasite, be able to discuss diffusion and osmosis, be able to identify the effects on a cell of isotonic, hypotonic, and hypertonic conditions, and be able to name 2 types of passive transport and 3 types of active transport. We start off this topic by looking at what nutrients microbes need. Not surprisingly, these needs are very similar to ours. However, microbes will obtain these nutrients in a very different way than us, and will also need a different amount of each nutrient compared to us. Anything that is needed by the organism is known as an essential nutrient. Within these essential nutrients, there are 2 subgroups, macro and micronutrients. Macronutrients are nutrients that are required in large amounts. Makes sense. As we learned about in the macromolecules topic, all biological molecules need carbon, so it's not surprising that carbon is a macronutrient. If an organism only needs a small amount of a substance, it's known as a micronutrient. One last note on chemical organization. If the compound contains carbon and hydrogen within the molecule, it is considered organic, whereas if it doesn't contain both carbon and hydrogen, it is considered inorganic. Make sure you have this concept straight, because it is different than how we use the terms in our colloquial or commonplace language. So now we know some things about the nutrients needed by the cell, but let's look at why. This is because the cell is in need of these nutrients to maintain life and to grow and divide. As I'm sure you are aware, most cells are mostly water. In our E. coli example on this slide, 70% of the cell is water. This is seconded only by proteins as a group of biological molecules. As you can see from the chart, 50% of the dry weight is composed of proteins. Dry weight is essentially anything left in the cell after the water is removed. As you can see, the cell is mostly composed of 6 elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. One last thing to note, the components are not brought into the cell as single atoms. Instead, they come in as compounds and molecules. One easy example is sodium and chloride. The cell can easily obtain this by ingesting sodium chloride, aka salt. Microbes are divided into 2 main categories based on carbon source. These are autotrophs or self-feeders and heterotrophs. Autotrophs can use carbon dioxide from the air to create the nutrients they need. Autotrophs that use sunlight to create these nutrients are known as photoautotrophs. These organisms can photosynthesize using sunlight and carbon dioxide. The other type of autotroph is the chemoautotroph. These organisms harvest the energy they need through a variety of really interesting pathways. Because they can do this, they don't need sunlight or other organic nutrients. Now let's turn our attention to the heterotrophs. Heterotrophs have to get their carbon from an organic source. This basically means they cannot use carbon dioxide as a carbon source. Photoheterotrophs can combine the use of sunlight and these organic carbon sources to obtain their energy. The main group of heterotrophs we need to focus on are the chemoheterotrophs. You and I are chemoheterotrophs. We get both our energy and our carbon from organic sources. Basically, we have to eat everything we need. Being in the sun doesn't do us any good. As far as microbes go, there are 2 types of chemoheterotrophs. saprobes, and parasites. Saprobes survive by obtaining nutrients from dead organisms. These are decomposers such as fungi and bacteria. Think of the microbes that work in composters. There are also parasites. These microbes love to live off of living hosts. These are all the lovely pathogenic bugs we like to talk about, as well as your normal microbiome. As long as their requirements are somewhat flexible, we can artificially culture them in the lab. If they are strict in their requirements, we have a difficult time culturing them. This is why it makes it difficult to determine the cause of disease from cultures on obligate parasites. Regardless of how these nutrients are obtained, there are 6 essential nutrients all living things need. I'm not going to read this table to you. Instead, make sure that you know the 6 nutrients, where they are found, and some examples of why they are important. So now that we know what cells need and how they get them, let's talk about how they actually get them into the cell. There are 2 main types of transport, passive and active. Passive means that energy is not needed to import the molecules into the cell, while, I'm sure you can guess, active means that energy is needed to get these nutrients into the cell. Let's start by looking at passive transport mechanisms. The first of these is diffusion. In this process, molecules automatically work to have an even distribution. This can occur across cell membranes. However, in order to move freely across the membrane, these have to be very small molecules. If the molecules are too large to diffuse across the membrane, they can pass through a protein in the membrane. This is known as facilitated diffusion. Remember, these 2 processes require that molecules only move from areas of higher concentration to lower concentration, with the goal of balancing the concentrations on either side of the membrane. If the cell wants to import something against the concentration gradient, or something very large, they need to use energy for this to happen. Let's look at these types of active transport. The first of these is carrier-mediated active transport. This is similar to the facilitated diffusion in that a protein helps carry these molecules across the membrane. However, in this case, since it is going against the concentration gradient, in other words, it's going from an area of low concentration to an area of higher concentration, the carrier must use ATP to help this transport occur. In group translocation, the cell uses energy to transform the molecule from one form to another while it is being transported through the membrane. This transport also occurs through the use of a protein in the membrane. The last process in the active transport involves bringing large items or large amounts of items into the cell. This is the process of phagocytosis and pinocytosis. Now remember, can this happen when a cell has a cell wall? No. This can only happen in cells that do not have a cell wall. Phagocytosis is known as cell eating because it moves solid items into the cell, while pinocytosis brings liquids into the cells. This process is known as cell drinking. To wrap up this topic, let's talk about how water moves in and out of the cell. Hopefully, you remember from the earlier slide that E. coli cells are 70% water. This means that cells need a way to ensure that they maintain the proper osmotic balance, or balance of water to salt or sugar concentration within the cell. Water can move freely across the cell membrane. This means that it is very important that cells control which environment they are in. For example, if the cell is in a hypotonic solution, the water will flow into the cell. This can cause the cell to burst if it doesn't have a cell wall. Cell walls are designed to help prevent this rupture. If the cell finds itself in a hypertonic solution, the water will flow out of the cell, causing it to shrivel up and die. Cells have a variety of mechanisms to prevent an imbalance, but you don't need to worry about this too much in this class. I've put some extra videos on osmosis in Blackboard if you would like to learn more about this concept. The big take-home message on this is that water balance is very important to the cell, and ensuring this balance is maintained is very important. So, to wrap up this recorded lecture and to remind you of what the learning objectives are, be able to list the essential nutrients of a bacterial cell, be able to differentiate between macronutrients and micronutrients, be able to construct 4 different terms that describe an organism's sources of carbon and energy, be able to define saprobe and parasite, be able to discuss diffusion and osmosis, be able to identify the effects on a cell of isotonic, hypotonic, and hypertonic conditions, and be able to name 2 types of passive transport and 3 types of active transport.